When you imagine a typical plant, you probably think of an organism that utilizes light from the sun and water from the soil to survive, grow, and reproduce. While most of us are familiar with some alternative plant strategies, such as carnivory, parasitic plants are another group of plants that redefine our understanding of how plants acquire and utilize key nutrients. Parasitic plants comprise of more than 4,000 species across the globe, making up a total of 1% of all flowering plant species. These plants connect to other plants via unique organs called haustoria, which act like a vacuum, extracting nutrients and water from the host to the parasite. Within parasitic plants, there's a wide range of diversity, spanning from fully parasitic endoparasitic plants like the corpse flower, the largest flower in the world, which grows entirely inside of a host plant until flowering, to partial root parasites or hemiparasites like the paintbrush flower, which are generalists that connect below ground to dominant species within plant communities.
My name is Jordan Argrett, and I'm a graduate student in the Odom School of Ecology studying the above and below ground interactions of root hemiparasites in natural communities. Root hemiparasitic plants are an important yet understudied group of plants that function as keystone species, modifying their biotic and abiotic environment.
In our current understanding, these plants parasitize competitively dominant species such as bunch grasses, reducing their size and competitive ability. This in turn facilitates the increase in either size or number of subordinate species, ultimately increasing local diversity. My research aimed to combine these two perspectives within the context of North American Castanea septentrionalis, a common perennial root hemiparasite in alpine meadow ecosystems in the Rocky Mountains of Colorado. Interestingly, several studies on this group have suggested that root hemiparasites might influence the community twofold. First, via the direct effect of parasitism on host plants, which we call the parasitism pathway, and second, via the litter pathway, where an increase in plant available nutrients are caused by the input of nutrient-rich litter from the parasite's tissue following the end of the growing season. By experimentally removing the presence of the parasite, and by adding litter to field plots, I aim to holistically examine the effects of root hemiparasites on the above ground and below ground interactions of plants, their associated microbes, and their environment. The results of my research will strengthen our understanding of how root hemiparasitic plants impact natural communities, facilitating our ability to tackle larger questions, such as the potential utilization as countermeasures to limit the spread of invasive species, the negative impact of parasitic agricultural pests on crop production, and the importance of their inclusion in the conservation of grassland ecosystems in North America. I would like to thank the ARCS Foundation for considering me as a finalist, and thank you all for allowing me to share my passion with you today.